Welcome and good morning to all B.Sc. first year students of Fishery Science. Boys, during last part we have studied the mechanism of respiration. Now today we have studied physiology of respiration. We have studying the important aspects respiratory system of Wallago. This respiratory system of Wallago, these are to be for our convenience divided into three parts. First, structure of gill. Second, mechanism of respiration, and third, physiology of respiration. These two parts we have seen last few parts. Then last part of respiratory system of Wallago are the physiology of respiration. Today we have seen how actual gaseous exchange is to be takes place inside the bronchial chamber or the gill chamber in fish body or in the fishes. How respiration these are to be takes place. This is to be we have seen physiology of respiration. The afferent bronchial artery, remember that fishes heart carry both pure and impure blood. Fish's heart comprises deoxygenated blood, and this deoxygenated blood these are to be passed from sinus venosus to auricle, auricle to ventricle, ventricle to bulbus arteriosus. Then this deoxygenated blood these are to be passed from bulbus arteriosus to ventral aorta, and ventral aorta these are to be divided, bifurcate on both sides, right side as well as left side, by four afferent bronchial arteries. And these afferent bronchial arteries these are to be carry the deoxygenated blood to supply to the bronchial chambers, to the gill chambers or the gills or the bronchioles. And this deoxygenated blood these are to be carried, these are to be bringing by afferent bronchial arteries. Physiology of respiration. The afferent bronchial artery bringing deoxygenated blood, afferent bronchial artery bringing deoxygenated blood breaks up into capillaries, into lamellae of gill filaments where exchange of gases occurs by osmosis through their thin wall. How the physiology of respiration these are to be takes place? The afferent bronchial artery bringing deoxygenated blood, this deoxygenated blood these are to be break up into capillaries, into lamellae of gill filaments, where exchange of gases occurs by osmosis through their thin wall. Osmosis, that is, in water more dissolved oxygen is to be present, and in blood more carbon dioxide is to be present. This carbon dioxide and oxygen which is to be present in water, and carbon dioxide and oxygen which is to be present in blood, these gaseous exchange, osmosis these are to be take place. Osmosis these are to be occur. What do you mean by osmosis? Lower concentration of is to be flow with higher concentration, and this osmosis these are to be takes place lower to higher, or both these are to be similar, because in blood lower concentration of dissolved oxygen to maintain the same level, dissolved oxygen level from water. These are to be absorbed, and in blood more carbon dioxide level, these are to be occur. This carbon dioxide, these are to be diffused into water. Such osmosis, these are to be take place by through thin lamellae of or thin walls of gill lamellae, and these gill lamellae, these are to be take in the gaseous exchange. The blood flowing in a capillaries, the blood flowing in a capillaries of the gill filaments gives up, capillaries of the gill filament gives up its carbon dioxide and absorbs oxygen from the water. The blood flowing in a capillaries of the gill filaments, these blood which is to present in the capillaries of the gill filament gives up its carbon dioxide and absorbs oxygen from the water. Means from the blood carbon dioxide, these are to be diffuses, and from water dissolved oxygen, these are to be absorbed. Dissolved oxygen, these are to be absorbed from water, and carbon dioxide which is to present in blood, these are to be diffused out, these are to be leave out. The efferent branchial artery carries away, remember afferent and efferent. Afferent always carry deoxygenated blood. The efferent branchial artery always carry oxygenated blood. Efferent branchial artery always carry away the oxygenated blood from gill to the body. During this process, the esophagus is kept closed by the esophageal valve to check the entry of water into it. Means when blood, these are to be purified, means from blood carbon dioxide, these are to be diffused, carbon dioxide, these are to be diffused, leave out, and from water dissolved oxygen, these are to be absorbed. Means blood, these are to be purified, more oxygen level, these are to be occur in blood. Such blood, these are to be oxygenated blood. Then this blood, these are to be passed, this blood, these are to be forward to all body parts by efferent branchial arteries. Efferent branchial arteries to efferent branchial arteries, then blood, these are to be passed to the lateral aorta, and then it is to be passed to the dorsal aorta. Lateral aorta, these are to be supply anterior part, head part, and dorsal aorta, these are to be supply whole body of the fish. This means these efferent branchial arteries, these are to be always carry oxygenated blood. During the process of gaseous exchange, during the process of exchange or the physiology of respiration, esophagus which is to be present in the mid region, in the midway, this esophagus, these are to be kept closed tight due to a esophageal valve, and when esophageal valve, these are to be working, these are to be function, at that time the valve, these are to be closed tight, and entry of water, these are to be checked, which is to be enter inside the mouth region. This is to be closed tight, only water, these are to be passed from mouth to the buccal cavity, pharynx, and then it is to be enter into the gill chamber or the branchial chamber, and in branchial chamber gaseous exchange, these are to be takes place. After gaseous exchange, water, these are to be expelled. Last period we have seen that make an inspiration and expiration, how it is to be occur, how it is to be seen. In this way, expiration and inspiration, these are to be takes place, and gaseous exchange, these are to be occurs in the branchial chamber or the gill chamber. The structure of the gills and the distribution of afferent and efferent vessels. The structure of the gills and the distribution of afferent and efferent vessel is such that the respiratory surface comes in close contact, with the respiratory water current, and when this structure of gills and distribution of afferent and efferent vessels, these are to be in contact with respiratory water current, and when respiratory water current, these are to be in contact, then gaseous exchange, these are to be takes place by gill filaments or the gill lamellae, and when gaseous exchange, these are to be takes place, gaseous exchange, these are to be occur in gill filament. These are to be dissolved oxygen, these are to be absorbed, and carbon dioxide, these are to be diffuse out, these are to be leave out. These are to be leave out or diffuse out. A high percentage of oxygen in the respiratory current, a high percentage of oxygen in the respiratory current, approximately 60% of it's absorbed by the gills. Those respiratory current, those oxygen which is to present in water, from this water, high percentage of oxygen which is to present in water which is to present in gill chamber or bronchial chamber, from this water approximately more than 60% of oxygen which is to be absorbed by the gill filaments or the gill lamellae. More than 60% of the oxygen, these are to be absorbed, these are to be used by the gill filaments or the gill lamellae, and hence blood, these are to be purified, respiration, these are to be takes place. The rate of oxygen consumption is related to the size of the fish. The rate of the oxygen consumption is related to the size of the fish and amount of oxygen present in water. That is, what is the rate of oxygen consumption is related to the size of the fish, and it is to be size of the fish, and it is to be depend upon the amount of oxygen present in water. Present in water means 2 points, 2 factors, these are to be important. One is the size of the fish, and second factor, that is the in water how much amount of oxygen these are to present. These 2 factors, these are to be determine how much quantity of dissolved oxygen is to be absorbed or consumed by the aquatic animal. The rate of consumption of oxygen in aquatic water bodies or the rate of consumption of oxygen by fish is to be depend upon the size of the fish and amount of oxygen present in water. The respiratory rate increases with the increase in carbon dioxide concentration. The respiratory rate increases with the increase in the carbon dioxide concentration of water in the environment of the fish. As how much respiratory current, how much respiratory rate these are to be occur, this respiratory rate these are to be increased, how much carbon dioxide concentration these are to present in water. Depending upon this carbon dioxide, more the carbon dioxide, more is the respiratory rate current. Less is the carbon dioxide, then less is the respiratory rate. Means depending upon the level of the carbon dioxide, that is respiratory rate increases or it is to be decreases. Increases when increase in carbon dioxide, decreases when it is to be decrease. Means in environment, more the carbon dioxide level in water, more will be the respiratory rate. Less the carbon dioxide, because more the carbon dioxide, more will be the oxygen these are to be used, because in that water when carbon dioxide is to be more, means ultimately oxygen these are to be less, and to fulfill the required quantity of oxygen they can absorb more oxygen. Hence respiratory current, respiratory rate these are to be increases depending upon the level of carbon dioxide. The respiratory cycle is controlled by the central nervous system. The respiratory cycle of fish, these are to be controlled. This respiratory cycle is to be controlled by the central nervous system. Means depending upon the central nervous system, the respiratory rate is to be decreased or it is to be increased. Totally it is to be controlled. Totally it is to be under the control conditions of the central nervous system. Thus, fish requires a constant supply of oxygen-rich water for the good health, for the good culture, good fish production. In those water, aquatic animals or fishes these are to present. In these water, fish require constant supply of oxygen. Those oxygen in which rich dissolved oxygen is to present, rich oxygen is to present, means the fish requires a constant supply of oxygen rich water for the respiration, for the gaseous exchange. If the water is depleted of oxygen, if the water is depleted of oxygen or exhausted, or fish removed from water, it soon dies. If the water is depleted or low oxygen level is present, complete oxygen is to be over from the water, or fish these are to be removed from the water for 5 minutes, 10 minutes. Soon it dies due to lack of oxygen, due to absence of dissolved oxygen, because these aquatic animals they can use only oxygen, but that oxygen is to be in dissolved state, not gaseous state like that of the human being. They can absorb the oxygen. That oxygen is only in liquid state, and when fish these are to be dies soon, that is called as the asphyxiation or the suffocation. Means fishes these are to be die when oxygen level these are to be decreased, or oxygen is to be completed over complete empty, no oxygen is to be present in water, or fish it can be removed from the water bodies for 5 minutes, 10 minutes, or for half an hour. Then due to the lack of dissolved oxygen, due to the lack of oxygen, fish can suffocate, and after suffocation it can be died, or asphyxiation these are to be takes place, and fishes are to be died. For that purpose, to avoid this asphyxiation or the suffocation, a rich dissolved oxygen water is to be supplied regularly for the fishes for respiration. In this way, gaseous exchange takes place in the branchial chamber or the gill chamber. With this, in gill chamber or branchial chamber, gaseous exchange these are to be takes place, and physiology of respiration these are to be performed. Physiology of respiration is nothing but the gaseous exchange. Dissolved oxygen is to be absorbed, and carbon dioxide from blood it is to be diffused out, leave out. Such gaseous exchange, such alteration of gases, dissolved oxygen is to be absorbed and carbon dioxide is to be leave out. Such alteration is called as the physiology of respiration. And with this we have completed respiratory system of Wallago. First we have seen structure of gills, second we have seen mechanism of respiration, and third that is the physiology of respiration. With this important article we have completed. Up to here we have seen important aspects of Wallago. Thank you all of you.